Hi everyone. First of all, I would like to thank the organizers and especially Aurora Pujol for inviting me. It's a pity that the meeting cannot be presential this time, but I'm pretty sure that we will have the opportunity to meet normally soon. My talk is about polygenic risk scores and potential applications in COVID-19. Polygenic risk score, derived from summary statistics of genome-wide association studies, GWAS, is a useful tool to infer an individual's genetic risk for health outcomes and has gained increasing popularity in human genetic research. In principle, a polygenic risk score is a weighted sum of allele counts across variants, where the weight reflects the magnitude of association between variant alleles and a trait or disease, as defined in a discovery genome-wide association study for the trait of interest. While the term polygenic score can apply to any phenotype, it is more commonly referred to as polygenic risk score, PRS, or genetic or genomic risk score, GRS, when describing disease risk. Development of a PRS is a multi-stage process that begins with testing in a risk model predicting the phenotype of interest, which may be combined with other non-clinical variables. Collectively, all variables included in the risk model are referred to as the risk model parameters. After fitting procedures to select the best risk model, the model is validated in an independent sample. The performance of a model is demonstrated through risk score distribution, discrimination, predictive ability, and calibration. Variants included in PRS construction are selected from a discovery cohort. Either whole genome sequencing or genotyping data can be used. Common variants and their effect sizes are typically selected from GWAS summary statistics. The summary statistics of a GWAS needs to undergo a variant selection or variant effects modeling to improve the predictive power of the PRS. The selection includes pruning and thresholding, Bayesian PGS, stacked clumping and thresholding, and metascoring. Currently, the gold standard for validation of the PRS, developed from the discovery cohort, is to validate against an independent dataset, ideally biobank scale or including diverse ancestries. The output of the validation phase is the selection of an optimal PRS with the highest performance in accurately predicting the trait of interest. Finally, the external validation phase involves computing the PRS in an independent set of individuals or population, unseen by the PRS construction process, and assessing its predictive power to confirm its predictive performance and to minimize the possibility of overfitting during the PRS construction. After a PRS for a specific trait or disease outcome has been established, tested, and validated, it can be used to calculate relative genetic risk for traits, diseases in new samples and cohorts. One promising application for PRS is integration as an additional risk factor in absolute risk models. For instance, the breast and ovarian analysis of disease incidence and current estimation algorithm is Boadicea. Reporting is also an important phase. The actual score for a specific trait can be communicated in several ways, depending on its intended application. In general, PRS results are normalized by scaling to a mean of 0 and standard deviation of 1 to facilitate interpretation. In addition to a PRS results, performance metrics of PRS should be reported that include the hazard ratio for a trait, the area under the receiver operating curve, AUC, goodness of fit measures, such as R-square, and the effect of a PRS in patient cohort can be also evaluated by calculating the net reclassification index to verify whether The addition of a new predictor to an absolute risk model results in differential classifications of individuals across thresholds. Currently, implementation of PRS in clinical setting is hampered by the lack of regulated standardized reporting. This challenge limits the use, interpretation and comparison of PRS, and some efforts, such as the Polygenic score catalog, are being done in this regard. Although PRS enjoys both simplicity and computational efficiency, yet the predictive performance of PRS remains moderate for most of diseases and traits. The reason is that genomic architecture and environmental and genetic interactions are more complex than expected. Despite these difficulties, the fact that the genome-wide polygenic scores for some common diseases identify individuals with risk equivalent to monogenic mutations, and that risk estimation is, for all intents and purposes, the earliest measurable contributor to common heritable disease risk, open expectations that genetic profiling could be considered a useful component of health management. Indeed, recent studies suggest that for a subset of diseases, our knowledge of the genetic factors underlying these conditions has improved to a point where polygenic risk profiling on the basis of calculated polygenic risk scores provides personal and clinical utility. Genetic architecture is often categorized as monogenic versus polygenic, meaning that gene perturbations contribute to the occurrence of disease in an individual, respectively. In reality, the genetic architecture of common adult-onset diseases is likely a continuum of common low-risk to rare high-risk genetic variants that can act together to drive overall risk in a single individual that can be captured with a variety of technologies. The increasing number of GWAS due to the establishment of large consortia and the increasingly decreasing cost and better performance of high-density arrays are open new possibilities to increase the accuracy of PRS approaches. Polygenic risk scores may detect high-risk individuals regarding COVID-19 susceptibility and severity. The actual susceptibility and severity of COVID-19 varies widely within the population. The left panel, redder individuals are more, greener individuals are less prone for severe COVID-19 disease. Genome-wide association studies might distinguish a group of SNPs from which a clinically relevant polygenic risk score can be built. This is in the right panel. Color-coded squares represent the presence of the risk allele in red or the alternative allele in green in each individual. The intensity of red corresponds to the odds ratios of the risk allele compared to the alternative allele. Resultant values of the odds ratios of each SNP are color-coded as the polygenic risk score in the orange background. Personalized risk scores correlates well with the actual COVID-19 risk. However, additional environmental, epidemiological, and comorbidities also modify the phenotype. The possibilities of generating polygenic risk scores for COVID susceptibility depends on the power of GWAS studies and the clinical and epidemiological data. The first study, published a little bit more than 1 year ago from an Italian-Spanish consortium who conducted a GWAS for severe COVID in around 1,600 patients and 2,000 controls, power enough to identify a region in chromosome 3, until now the most important associated region for severe COVID in both odds ratios and p-values. Chromosome 3, the affected area, includes several actors which might alter COVID-19 susceptibility, including chemokine receptors, while in chromosome 9, the association signal coincided with the ABO blood group locus. ABO blood group has independently been linked to COVID-19 in some studies. Interestingly, a study carried out by Hugo Sever and Svante Paavo shown that the associated chromosome 3 region was inherited from Neanderthals, and this has different implications from an evolutionary and population genetic point of view. One of the most important GWAS studies carried out until now was the one published by Castiñeiras et al. In Nature last December. They recruited more than 2,000 critical ill patients from more than 200 intensive care units in Europe, and as a control they used the 100,000 Genomes project. They found 3 novel hits. In addition to the chromosome 3 cluster, they found to be involved the gene encoding dipeptidyl peptidase 9, DPP9, and especially important were the signal in chromosome 12 with the SNPs in the gene cluster encoding antiviral restriction enzyme activators, the OAS family, and even more important, because it's a potential target for drug repurposing, the interferon receptor gene IFNAR2 in chromosome 21. The huge effort of the Host Genetics Initiative produced the most important GWAS up to date. It was the result of 3 genome-wide association meta-analyses comprising almost 50,000 COVID-19 patients from 46 studies across 19 countries. They include more than 7,000 hospitalized patients and more than 6,000 critically ill patients in hospital. And especially they have enormous power because they use 2 million controls, including the 23andMe cohort. They reported 13 genome-wide significant loci that are associated with SARS-CoV-2 infection of severe manifestations of COVID-19. Several of these loci correspond to previously documented associations to lung or autoimmune and inflammatory diseases. They also represent potentially actionable mechanisms in response to infection. Always in close contact with the HGI initiative, we set up a consortium that we have called SCOURGE. Most of the samples are from hospitals in Spain, with a total of around 17,000 samples, including the samples from the consortium Stop COVID. From different hospitals and institutions from some Latin American countries, we have collected additional 8,000 samples. We have also collected a detailed clinical data with 174 variables and required a minimum number of 17 variables to enter data. And additionally, we have performed a rigorous quality control of the clinical data. With this data, we automatically classify the severity of the disease in 5 groups that are quite well equilibrated, the same as the distribution by sex. For the GWAS, we use the Spanish biobank array in more than 11,000 cases and 6,000 controls of general populations in samples from Spain and around 3,000 cases from Latin America. For replications, we have used an independent cohort with almost 1,600 COVID-19 cases from 6 different Spanish biobanks and 1,500 controls. After a standard quality control, we have analyzed the results for both European ancestry and non-European ancestry. One of the advantages of this study is that cases and controls in Spain are from the same population, so avoiding the confounding effects of population stratification. As it can be seen in the Manhattan plot, the comparison of hospitalized versus rest of cases, asymptomatic and controls, show clear signals in chromosome 3 region and chromosome 21 interferon receptor. Most of the hits of the HGI study, but not all, replicates in the same direction. Again, SNPs in genes affecting viral entry, innate immunity, or inflammatory pathways are involved. Top hits were not validated in the Latin American cohort with major non-European ancestry, pointing to important differences among populations, although sample size is still low, so we are recruiting and genotyping more cases and controls from Latin American cohorts. Genetic risk scores created using the list of SNPs reported as associated with hospitalization severity of risk in the meta-analysis performed by the HGI. Genetic risk scores was calculated for each patient of the scourge cohort belonging to any severity class and also for control for general population. To evaluate the existence of genetic risk differences along the complete proposed severity scale, the genetic risk score mean value was compared between the 6 classes, 5 severity levels plus controls from general populations via ANOVA. GRS analysis outlined the existence of 3 severity categories in our data as opposed to the initial 5-level scales clinically defined. The GRS differentiated 3 main groups. For these purposes, we used the multinomial model to test the associated of a 3-level severity scale, control plus asymptomatic plus mild severity level, intermediate plus severe cases, and very severe cases, with the clumping SNPs that reached known-wide significance in the meta-analysis. Similar results were found in this study of Horowitz et al. From Regeneron. To my knowledge, still in preprint in meta-archives, but quite relevant, so I feel obliged to mention it here. This is a large GWAS in more than 750,000 individuals that provides the first genetic evidence that rare variants in ACE2 expression influences COVID-19 risk, and also yields genetic risk scores predictive of severe disease that are shown in this slide. In A, association between high risk and hospitalization. Risk of hospitalization among cases is shown for individuals in the top genetic risk score percentile, agnostic to the number of clinical risk factor present. The association was tested in 3 studies using logistic regression with established risk factors for COVID-19, included as covariates. Results were then meta-analyzed across studies from a combined samples of around 45,000 COVID-19 cases, including around 6,000 hospitalized. In B, can be seen the association between high genetic risk and severe disease using the same approach for 45,000 cases and near 2,000 severe cases. This slide shows the comparison of effect sizes across COVID-19 susceptibility and severity outcomes for 6 previously reported risk variants that were validated in the Regeneron study. The same as in our study, the SNP in LZ-TFL1 has the highest power to predict severity. This slide shows the association between the 6 SNP genetic risk score and risk of severe disease among COVID-19 cases of European ancestry after stratifying by the presence of clinical risk factors. High clinical risk was defined as any one of the following: age major than 65 years old, BMI bigger than 35, chronic kidney disease, diabetes, or immunosuppressive disease, age more than 55 and presence of chronic obstructive pulmonary disease, or the chronic respiratory diseases, cardiovascular disease, or hypertension. The results are quite impressive, and this is a clear example that a genetic risk score based on common variants validated in this study can be used to identify individuals at high risk of poor disease outcomes and that could have a potential clinical utility, especially if combined with clinical risk factors. A similar approach was used in this recently published study where a PRS with 112 SNPs in more than 400,000 participants from the UK Biobank study was constructed. High genetic risk correlates with a higher risk of severe Covid-19, regardless of pre-existing COPD. Although severity is associated with COPD, the risk of incident severe Covid-19 at high genetic risk combined with pre-existing COPD was 2.05-fold higher than at low genetic risk and without pre-existing COPD. The risk of incident severe Covid-19 at high genetic risk was 1.5-fold higher than that at low genetic risk, suggesting that PRS potentially was suitable to the risk stratification among those with Covid-19. Finally, ROC curves were constructed and the area under the curve was estimated for different combinations, age, sex, comorbidities, and PRS, given the best result. Other GWAS where polygenic risk scores were calculated is this one from the CCPM with 36,000 cases in admixed populations. As expected, PRS distributions differ by ancestry, pointing to important population differences, which makes sense since many of the genes involved in COVID are probably under positive selection pressure due to different gene-pathogen interactions that differ among populations. The same was observed in this study from India. 100 SNPs selected from the Castañedas et al. New England study were selected, and a positive correlation between polygenic risk scores with mortality and severity was found. They used a p-value cutoff of 10 to the minus 6 derived from the previous GWAS summary statistics. Selected SNPs overlapping with the India Genome Variation Consortium and with similar frequencies in the Indian populations were selected. They found that Indian subpopulations differ with respect to the genetic risk for developing critical illness and suggest the use of population risk scores for more efficient vaccination schemes. Polygenic risk scores can be used to determine common genetic background among diseases by using a discovery sample and a target sample. For instance, in this study from our group, polygenic risk scores were obtained from sequencing the data of almost 400 Spanish autism trios using as discovery sample GWAS summary statistics from the PGC repository. By using polygenic transmission disequilibrium test, we found that the risk for schizophrenia is overtransmitted to children with Asperger, but not to non-Asperger autism cases. The results found in this study supports the idea of Asperger being qualitatively distinct from non-Asperger autism and consistently evidence that the genetic overlap between Asperger and ADHD, MDD, and schizophrenia. This can be applied to see interactions or common genetic background between COVID severity and other pathologies. We are currently exploring some of them, with negative results in the case of COVID and Alzheimer's disease, where the analysis of GWAS data from our Alzheimer's disease, the GESCO cohort, didn't show any involvement of the ApoE locus in the COVID cases, nor a common genetic background. However, for some other diseases, comorbidities contributing to severity, we are obtaining promising results. Just to conclude, polygenic scores have become powerful tools for genetic disease research, genetic testing, and clinical trials. Development of polygenic risk scores begins with initial construction, followed by validation and testing to determine the optimal polygenic risk score. It can then be used to calculate relative genetic risk for diseases in new samples and cohorts. As multiple GWAS on host genetic susceptibility are ongoing, several genomic susceptibility loci are being consistently replicated. In addition to viral entry and innate immunity, other mechanisms, including adaptive immunity, are also of paramount importance regarding the susceptibility to COVID-19. Translating these individual susceptibility variants into clinical relevant polygenic risk scores could fully leverage this acquired knowledge to detect high-risk individuals and to fulfill the requirements for polygenic risk score validation. Combination with clinical and epidemiological data will be key for translation. To better characterize putative genomic susceptibility loci, well-designed international genome-wide association studies are needed, especially in non-European populations, since important population differences are expected. And that's all. But before finishing, I would like to thank all my team. I am very, very proud of all of them, and they made an impressive work in this COVID-19 US study. But of course, I have to thank all collaborators in the SCORCH Consortium, especially Pablo Lapuncina, the Stop COVID Consortium, the CENIO Consortium, the HGI Consortium, as well as the funding agencies, Instituto de Salud Carlos III, and charities, Fundación Amancio Ortega and Banco de Santander, and all the healthcare personnel that helped not only with this project, but with everyone in these difficult pandemic times. Thank you so much for your attention.